Dr. Pettigrew, can you explain what is a biomarker and why is it important?
A biomarker is an observable feature or parameter of a disease or condition that tells you something about the state of that disease or condition. They're very useful in determining the presence of disease. They're useful in assessing the state of the disease and characterizing the level of the disease, and also in monitoring therapy and the response to therapy.
You like to say that you're director of the National Institute of Cool Stuff.
Indeed.
One example is these fluorescent markers that might be used during surgery. Can you talk about what they do?
This is the brainchild of a Nobel laureate, Roderic at UC San Diego, and one of his research fellows, Gwen Nguyen, also at UC San Diego, head and neck surgeon. I may have mispronounced Gwen's name. Forgive me if I've done that. But these two have teamed up to solve a challenge in surgery that the public might not really appreciate really exists. And that is exactly, where is the tumor? Where does it begin? Where does it end? What are the margins of the tumor? How deep, how far, how wide, etc. Easy to see with some of our most sophisticated images, like MR images, before you go in the operating room. Often difficult to see in the operating theater, where you don't have the advantage of these sophisticated three-dimensional imaging technologies. So what Gwen and Roger have teamed up to do is to leverage his expertise in fluorescent proteins, which is what he shared the Nobel Prize for, and develop a marker that fluoresces only when it attaches to and enters a tumor cell. It makes the tumor cell glow brightly with a color that anyone can see in the operating room, so that you can identify not only the presence of the tumor, but exactly where the margins are at a cellular level. This gives the surgeon an easy to follow roadmap of where to cut and where not to cut. That's advantage one. The second advantage is that you can do the same process to identify the nerves. And the nerves that may invaginate the tumor, encase it, go under it, through it, around it, often difficult to see in the operating room. It can be tricky to follow and find. And you've heard from time to time where operations occur and there was accidental cutting, a nicking of a nerve. It's a particular challenge in the head and neck area where there are many nerves and the tumors can be quite large. And it's also a challenge in prostate surgery, where the removal of the prostate often results in some neurological deficit. Being able to identify the nerves separately from the tumors and separate those two structures out so that the surgeon can carefully and precisely remove the tumor, leaving the nerves unharmed, is the goal and the promise of this exciting, cool technology.
How soon might we see this in our local hospital?
So these two have been working on this for a few years now, and they are progressing along. They've done a number of animal studies and are planning to begin clinical trials in 2015, is my understanding.
Can you give an example of a medical moonshot?
Well, one of them was the one that I talked about today, and that is improving our ability to detect cancer early, non-invasively with a routine blood test by identifying the presence of rare circulating tumor cells. Doing this at a sensitivity of 1 tumor cell present per billion normal cells was the goal. And the research team that we funded with one of these quantum programs was a team headed by Mehmet Toner, an engineer at MIT and Harvard, and Daniel Haber, who is a cancer geneticist at MGH. Those two have been working for 7 or so years in order to achieve this medical moonshot of being able to take an individual's routine blood sample and determine whether or not you have early stage cancer as evidenced by the presence of circulating tumor cells, or even in patients who are known to have a primary cancer that is confined to the primary site, to see if it has begun to leak cancer cells as a prodrome to developing cancer metastases. This would be a tremendous advance because 90% of cancer deaths are due to metastatic spread as opposed to the primary tumor itself. And that was the initial goal. One of the wonderful things about pursuing medical moonshots and supporting scientific innovation and technological innovation is that one discovery leads to another great discovery and another pathway and opportunity for even more great discoveries. And that indeed happened here. So while the initial goal was to try and prevent metastatic tumor spread, what they have learned is by capturing these rare cells also gives you an opportunity to be more precise in characterizing the biology of the tumor, studying the genetic profiles of the cancer cells in an individual patient, and also the responsiveness of those cancer cells to different therapeutic drug possibilities, and determine which of those is most effective in treating that cancer. Following it along in time with a system like this that is so easy to do and so non-invasive allows you to continue to monitor the effectiveness of the therapy and determine whether or not the tumor is changing in such a way that it's no longer as effectively treated by the drugs that you started out with, but could be treated more effectively down the road by a different set of drugs. That is particularly exciting.
Dr. Pettigrew, thank you very much.
My pleasure. Thank you.